Okay, so welcome back to this next video in which we are discussing Pavlovian/classical conditioning. Okay, so we're discussing Pavlovian conditioning now in a species of sea slug known as Aplysia californica, also called the Californian sea slug. Okay, right, so I've told you that we're going to see this form of learning in the Gill withdrawal reflex of the Aplysia californica. Okay, and just to remind you, the Gill withdrawal reflex involves dropping water onto the siphon on the back of the Aplysia californica and watching the gill, which is this flap of muscle, contract and cover over the siphon, basically. Okay, right, what we firstly want to see is what actually is involved in this Gill withdrawal reflex. What is the neural circuitry involved here? Okay, then what we want to do is we want to talk about sensitization firstly. I told you we were going to have to discuss sensitization before we discuss the Pavlovian conditioning. If we discuss the sensitization of this Gill withdrawal reflex, we will be in a very good position to understand the Pavlovian conditioning, because Pavlovian conditioning in this system is just a slight modification of that. Okay, right, so let's begin with the neural correlate of this Gill withdrawal reflex, and it's fantastically simple. This is why neuroscientists will love this so much. Okay, so basically, there is a ganglion, okay, within the Aplysia californica, which sits down here, or there around, and the siphon and the gill are up here, okay, and this ganglion contains lots of neuronal cell bodies, and this is called the abdominal ganglion of the Aplysia californica. Okay, and basically what happens is there is a single sensory neuron that innervates the siphon of the Aplysia californica. This is why it's just absolutely beautiful. There is a single sensory neuron that innervates the siphon. Okay, so I'll draw this here. So I'm going to separate out the little bits and have the neurons going from each portion. So here's the siphon here, and basically you'll have your sensory neuron coming from the siphon, and basically it will then have its cell body within this abdominal ganglion. Okay, so I'll draw the abdominal ganglion rather large. Okay, so here's the cell body of this sensory neuron, and the sensory neuron which innervates the siphon is called the L1, and then I'll put SN for sensory neuron. So just to remind you, SN stands for sensory neuron. Right, so let's put a bit of colour onto this to try and make it as simple as possible. Okay, and the sensory neuron is going to synapse onto the next neuron along. Okay, so we'll colour in the sensory neuron in green here. Okay, and it innervates the siphon down there. Okay, so that's the sensory neuron, and it will then innervate the next neuron along in this Gill withdrawal reflex. Okay, and the next neuron along is called the L7 motor neuron. Okay, so what will happen is the L1 sensory neuron will synapse onto this L1 motor neuron, which also has its cell body within the abdominal ganglion, and then the L1 motor neuron will leave the abdominal ganglion, and I'll just put an arrow from there to there, and it will then innervate the gill. Okay, so I'll draw the gill muscle here. We'll colour that in red. So there's the gill, and I'll colour in the motor neuron in orange. So this is the L7 motor neuron. It's the motor neuron which innervates the gill within Aplysia californica. Okay, right. So, very simple reflex system then now. Dropping water onto the siphon will activate the L1 sensory neuron. An action potential, or multiple action potentials of course, will fire along the sensory neuron and will trigger action potentials in the L7 motor neuron, which will come to the gill here and cause contraction of the gill. Okay, that's the Gill withdrawal reflex. And basically we can see therefore that it is a monosynaptic reflex. Okay, right. So, what I now want to discuss with you is sensitization of this Gill withdrawal reflex. Okay, because, as I say, sensitization, if we understand the sensitization, we will then be in a fantastic position to later understand the Pavlovian conditioning. Okay, right. So, sensitization then. So I firstly just want to define for you what sensitization actually is. So I'll start with a common sense example of what sensitization is. Okay, so basically, if you are walking down some lovely country lane and it's a beautiful spring day, let's say, and actually no, let's say you're walking down some pavement, okay, and it's a beautiful spring day, it's really light, and you hear footsteps behind you. What's your response? You just keep walking on. You ignore them. Okay, let's now say you are walking at midnight. It is pitch black dark. You hear footsteps behind you. What's your response then? It's the startle response generally. Okay, so your response to the same sensory stimulus is completely different depending on the scenarios in which you are in. So when you are in the pitch black situation at midnight, your response to the footsteps, the same sensory stimulus you heard earlier that day when you were in broad daylight, is completely different. That is a form of learning. The ability to adapt your response, to change your response to the same stimulus from information, other information that you have around you. That is the concept of sensitization, and it's a very basic form of learning. Okay, so basically the idea is that you can increase your response to a stimulus. So if you've got a stimulus, it evokes a response. Basically, you can increase the level of response to a stimulus from other information, okay? So from other stimuli, such as the stimulus that it's pitch black and dark. Okay, so a stimulus 2, I'll say. So basically, this is a form of learning. It might not be your typical idea of learning. I mean, generally, humans' typical idea of learning is sitting down and cramming fact, being able to recite who the monarch of England was in 1652 or whatever. Okay, that's what people generally view as learning. But in a broader sort of concept of learning, this is a form of learning. It's changing your response to a stimulus because of other information that you are receiving, basically. Evolving, that's what learning is about. Okay, right. Or adapting, maybe. Okay, so basically, we are going to see the sensitization of this Gill withdrawal reflex in response to electrically shocking the head of the sea slug. So basically, this is what they do. They electrically shock the head of the sea slug. So they deliver a little electric shock to the head of the sea slug. So here is my lightning bolt going into the head of the sea slug. Okay, and basically, when you deliver an electrical shock to the head of the sea slug, what you notice is that if you then repeat the experiment, i.e. you drop a water droplet onto the siphon and look at how much contraction you get of the gill, okay, i.e. you look at the gill withdrawal reflex, you find that the gill withdrawal reflex is hugely exaggerated after you've delivered the electric shock. So basically you see that the sea slug will initiate a much bigger gill withdrawal reflex if you have just given it an electric shock to the head, basically. So, this is sensitization. Our stimulus here is the water being dropped on the siphon. Our response is the gill contracting. Our stimulus 2, okay, analogous to the darkness, okay, is basically delivering an electrical shock to the head of the Aplysia californica. Okay, so delivering this shock causes you to initiate a bigger response to the same stimulus. So you have sensitized the Gill withdrawal reflex. What we now want to talk through is the actual neural correlate of this. Okay, right. So basically there is another neuron within the abdominal ganglion of the Aplysia californica. Okay, and this is known as the L29 interneuron. So this is the L29 interneuron. And I think I'll colour the L29 interneuron in blue. Okay, and basically the L29 interneuron will become active once you've delivered an electric shock. So the electric shock causes the activation of the L29 interneuron. Of course, there'll be other portions of a pathway prior to this. So, there'll be sensory nerves in the head, they'll activate a whole cascade of other neuron activation, and the overall result of this, for our purposes, is that this L29 interneuron within the abdominal ganglion is going to become activated, and I should just add the abdominal ganglions around there on this picture. Okay, so the L29 interneuron is going to become activated. Okay, so we've reduced all of the complexity of how the electric shock actually is perceived by the Aplysia californica and what pathways that triggers, to just leading to the activation of the L29 interneuron. Okay, so we don't care about the pathways in between here. And basically this L29 interneuron, it has an axo-axonic synapse. Okay, it synapses onto the axon terminal of the L1 sensory neuron, which then synapses onto the L7 motor neuron. Okay, so this is called an axo-axonic synapse. So when an axon synapses onto the axon terminal of another axon, that's called an axo-axonic synapse. So this is an example of an axo-axonic synapse. Okay, so the electric shock will cause the activation of the L29 interneuron. It will then release neurotransmitter onto the axon terminal of the L1 sensory neuron. So let me draw a little bit of a bigger picture here. So let's say this is the axon terminal of the L1 sensory neuron. Okay, then basically, well actually I'll draw, you'll have some dendritic spine basically of the L7 motor neuron here. So this is the L7 motor neuron here. Okay, and now you've also got the axon terminal of the L29 interneuron here. Okay, and it's synapsing onto the axon terminal of the L1 sensory neuron. So let's just color code these again. So L29 was in blue here. The L1 sensory neuron was in green here. Okay, and then the L7 motor neuron is in orange. Okay, right. So, what is the result of this? Well, basically, L29 is going to release the neurotransmitter serotonin, which we'll discuss more in a moment, okay, onto the L1 sensory neuron's axon terminal. And what that triggers is it triggers pathways within the axon terminal of the L1 sensory neuron, which will lead to the L1 sensory neuron releasing more neurotransmitter every time an action potential comes along. Okay, so let me repeat that because it's worth repeating. The L29 the L29 interneuron will release serotonin, also called 5-hydroxytryptamine, onto the L1 sensory neuron's axon terminal. This will trigger whole signaling cascades within the axon terminal of the L1 sensory neuron, which will then trigger, whenever the L1 sensory neuron fires an action potential again, it will cause the axon terminal to release a greater amount of neurotransmitter every time an action potential arrives. So, what does that now mean? Well, if we now squirt water onto the siphon of the Aplysia californica, the L1 sensory neuron will start firing, and now because the L29 interneuron has released this serotonin onto the L1 sensory neuron, what will now happen is the L1 sensory neuron will release a greater amount of neurotransmitter than it would usually, which will mean that L7 motor neuron fires action potentials more frequently, and that will cause a greater contraction of the gill. Okay, so because of the greater concentration of neurotransmitter that's being released onto the L7 motor neuron, you'll get a greater frequency of action potentials being sent down the L7 motor neuron. Remember, an action potential is an action potential. Stimulus intensity, which in this case is a neurotransmitter, does not change the size of an action potential. It changes the frequency of action potentials. You have to code the intensity of a stimulus by the frequency of action potentials, not the intensity of an action potential. Okay, you can't vary the intensity of an action potential. It's an on or off thing, binary. Okay, right. So that is the neural basis of the Gill withdrawal reflex. Now we need to discuss this in more detail actually. We need to see how serotonin is actually going to trigger the L1 sensory neuron to release a greater amount of neurotransmitter, because it is going to be important, because basically Pavlovian conditioning is going to involve a slight modification to this pathway, which will lead to a different overall outcome. So we need to look at the pathway. Okay, so I think I will call it there for this video, and in the next video we will begin discussing the pathway by which serotonin being released onto this L1 sensory neuron is going to trigger that L1 sensory neuron to release a greater amount of neurotransmitter.